Hello and welcome everyone to CyVerse Learning Institute's Focus Forum webinar series. I'm Tina Lee, CyVerse's User Engagement Officer, and we are thrilled today to kick off our fall series with a presentation on RNA-Seq basics, generating and processing raw sequence data using Nanopore technology and CyVerse tools. Today's webinar will be presented by Andrew Nelson, currently at the University of Arizona, and very soon to be of the Boyce Thompson Institute at Cornell. For those unfamiliar with CyVerse, we are an NSF-funded cyber infrastructure project, and this free webinar series is designed to fulfill part of our mission to train the next generation of scientists on how to use CyVerse's computational resources. So let's quickly take care of some housekeeping, and then we'll get on with the webinar. Today's presentation is roughly 30 minutes with time for question and answers at the end. Please open the chat window and type your questions for Andrew in the chat window, and we'll address those when he's done. Materials from today's webinar, such as the presentation slides and the video recording, will be posted on a wiki page for which we'll email you the link later today. The CyVerse Learning Institute is busy planning our second foundational open science skills course, and we're also planning our third container camp, both of which are scheduled for spring 2020. Please visit our website for more information on these trainings and to register. And so without further ado, I'd like to introduce Andrew Nelson. Welcome, Andrew. Please feel free to give yourself a better introduction than I just did.
Hi, Tina. Thank you so much for that very nice introduction. And thank all of you for attending this CyVerse webinar on RNA sequencing with Nanopore. So just to do a little bit of introduction, I like to call myself a CyVerse power user. I've been using CyVerse cyber infrastructure for a number of years now, probably more than I'd like to admit. And if I'm describing myself, the little keywords that you put down at manuscripts or when you're a reviewer or something, my keywords would be plants. I'm primarily a plant biologist. Transcriptomics, I do a lot of analyses around transcriptomics, comparative genomics, transcriptional regulation, stress biology, long night coding RNAs. And if you have questions about any of these topics, I would love to chat your ear off ad nauseum after this webinar is over. Okay, so some goals for today. I would like to briefly introduce all of you to Nanopore mRNA sequencing. It's an amazing technology. We're getting a lot of really interesting information out of this technology. Along with PacBio sequencing, we're finally getting really good coverage of full-length transcripts, and we're getting a better understanding of what is going on with those transcripts, isoform discovery, et cetera. I would also like primarily what we're here to talk to you about today is to familiarize you with the tools available in the discovery environment that will allow you to perform a basic workflow to analyze Oxford Nanopore Technology sequencing data, going from kind of quality controlled sequence to differential expression analyses, or kind of sanity checking and visualizing your sequencing data in a genome browser. Okay, but before we begin, some resources. As Tina said, some of this stuff is going to be sent around in an email later. I'm going to read the docs format. But I cannot stress enough, for those of you who have never done Nanopore sequencing, as soon as you buy your first kit, you will have access to the Nanopore community. And this community is essential. It provides all the protocols, and these protocols are laid out in really straightforward and easy to follow ways. And not only do they provide the protocols, but there are other scientists just like you and me who are commenting in this community. So this is a screenshot of their kind of introduction, or their landing page on their website after you log in. And if you go to the community, this will take you to where other scientists are posting questions, posting comments, providing their expertise for people who may have difficult to process samples, or who are just simply having trouble getting their sequencing to work for one reason or another. In addition, you can always feel free to email me later. Here's my email address. All of what we're going to be doing today, or what I'm going to be discussing, can be accomplished inside CyVerse discovery environment, and the read the docs that will be sent out later on today, as Tina was talking about. Okay, so nanopore sequencing. The MinION, the Minion, is super cheap. So a lot of you are probably going to be coming into or introducing yourself to nanopore sequencing technology using the MinION. And of course, you have to include this clever little, I have to include, because of my sense of humor, this clever little Minion. But of course, we're not going to be using it for sequencing. We're going to be using instead this little Minion instead, which is quite small, as Nanopore likes to advertise. It's essentially slightly bigger than a flash drive. It's a clunky flash drive, but it's still a very small device. And this flow cell right here, where you would actually load your sequencing library, contains, just in this little region here, contains hundreds of thousands of pores that are going to be actively sequencing your sample. So it's an amazing technology, and I'm not going to belabor it too much, otherwise I'll sound like a fanboy. But I would just remind you to go to Oxford Nanopore's website, look at their kind of demos, and check one out for yourself, because it's extremely cheap. The cost of entry is really low. So in terms of mRNA sequencing, what is Oxford Nanopore providing us? Well, the mRNA sequencing that we get, that we can perform with ONT, gives us kind of the following advantages over your standard Illumina short read or other short read sequencing. As I already alluded to, we get full length stranded sequencing, super informative, because you're no longer having to rely on computational algorithms to stretch across introns and kind of make guesses as to, I mean, not guesses, but we're getting complete coverage of a transcript, and we're able to delineate between isoforms in a much cleaner fashion. So we're getting better isoform resolution. And in addition, something that I'm particularly interested in is we're better able to identify antisense transcripts. Before, when you had short read sequencing data, identifying these antisense transcripts was a little bit challenging. But now, since we're getting the full length transcript and we always know which direction it's sequenced in, I remember hearing for both human and Arabidopsis, and I think chicken, there are roughly 30 to 50% of protein coding genes actually have antisense transcripts overlapping them. So they're performing some kind of important regulatory mechanism that we haven't really been able to visualize and kind of probe until now. So this is a pretty exciting information that we're getting from Oxford Nanopore sequencing. In addition, we are now, with Oxford Nanopore's recent kind of iteration of their technology, we're now able to identify post-transcriptional base modifications that are occurring on your transcripts without having to employ special antibodies like M6A sequencing, et cetera, or special detection algorithms. We see those base modifications occurring, and we can sequence them using ONT, using this little guy right here. And then, just like short read sequencing, Oxford Nanopore Technology is providing you the same type of, the same ability to perform differential expression analysis and to identify novel genes without a genome. So we are getting a whole bunch of, we're able to ask a wide variety of questions. Importantly, Oxford Nanopore Technology sequencing is fast, it's relatively cheap, and you can do it all yourself. It is this, basically, this technology is democratizing data discovery. So what do we do with all this power? How do we go from base called sequencing data that's coming off of this minion and turn it into something meaningful. And that's what we're going to be talking about today. So a general workflow, obviously, if you're in the lab, you have to extract RNA, you have to perform library preps, sequence, and then base call. We're not going to talk about any of this stuff today unless you have questions, and we can talk about it afterwards. Again, the Oxford Nanopore website, the community protocol page has really in-depth and kind of animated protocols for doing a lot of this. I have some tips and some advice for library preps and base calling just to make things go a little bit more smooth. But in general, all of this is very straightforward and is taken care of you. Oxford is doing a good job of taking care of these for you. But instead, what we're going to be talking about today is kind of the data processing and analysis and how you can do all of this inside of CyVerse's discovery environment, which is very intuitive. For those of you who have never used the discovery environment, very intuitive, GUI-based or graphical user interface platform where all the common kind of command line tools that you would use normally to process nanopore or short-read data, you can do in a very graphically intuitive way. Okay, so this workflow to process the data, and I'm going to talk about all of this in general, but there actually aren't very many steps to this. It's really easy and relatively fast and painless to get from sequence data to either differential expression tests of using your favorite algorithm, or to visualize your data in a genome browser. And today we'll talk about EpicCoG. So because I'm visually oriented and I hate lists, I like workflows a little bit better. Showing this to you in a different way, and showing you the algorithms or the kind of the tools that are installed in the discovery environment that you can use to proceed from grabbing a transcript FASTA file that you're going to map all your reads back to, all the way to performing differential expression tests using RStudio, using CyVerse's new Vice apps. So using RStudio in the cloud, so you don't have to download any data to your computer, and then visualizing your mapped reads also in the cloud on the EpicCoG platform. And so all of this, it's 6 steps, 5 steps really, if you don't even want to visualize. And that's 5 steps to success: science, nature papers, or the Ig Nobel Prize, your choice. I should point out that all of these tools that you, again, would use in the command line, if you were so inclined, all of these tools have been installed, loaded into the discovery environment by a group of really important people, the science analysts at CyVerse. And so if you see these names next to an app that's been installed, you should reach out and thank those people, because they are saints. Okay, so enough of that. Let's start by taking all of your data and getting it into CyVerse's discovery environment. So if you are performing nanopore sequencing, you are going to get out. It kind of varies. You're either going to get out, you're going to be performing base calling as the sequencing is occurring, in which case you're going to be getting FASTQ sequences from automatically from your sequencing, and that's what you are going to upload to CyVerse. Or you're going to be getting what's called a FAST5 sequence, which FAST5 file, which actually has all this information that will allow base calling downstream. So this is if you're performing sequencing and perhaps your computer doesn't have the requirements to do the memory requirements to perform base calling and sequencing at the same time. It's not a big deal. You can easily perform a base calling downstream on your own computer at a later date using Oxford Nanopore's kind of proprietary software, Guppy. It's the latest iteration or the improvement over Albacore, which was their first kind of base calling platform. And if you're just using if you're just performing mRNA sequencing, you can perform Guppy, use Guppy to do base calling on a standard standard PC, Mac, or Linux platform, and it probably won't take a super long time. If you're trying to do Guppy-based calling on genome sequences, then you should probably seek out some kind of a GPU platform, because otherwise it's going to take you weeks to base call your data. Okay, so what I'm getting at is that you're going to have FASTQ, so FASTA sequence plus the quality scores for your sequence. So those FASTQ files need to be uploaded. It's really easy to upload files to the discovery environment. This can either occur using the program CyVerse.org or using a direct upload option within the discovery environment. And we'll get to kind of the visuals. We'll switch screens here in a moment and get into the discovery environment. But again, if you have large files, I would recommend using CyVerse.org. It's very fast. And in addition, if you were doing your sequencing on a genome or a species, sorry, that isn't maybe it's a specialized species, it's not a commonly studied species, then you'll probably want to bring in your own genome and genome annotation file, which you'll also want to upload using CyVerse.org, most likely. Okay, so the very next step, once you have your files uploaded into this discovery environment, you're going to want to create a transcript file from your genome of interest. So this is going to be a FASTA file of the sequences of just the known genes in your genome. And you can easily do that using the package or the program GFFread, which I've highlighted here. And the GFFread package or program comes out of the cufflinks package from the Trapnell lab. So the inputs for GFFread are simply the FASTA genome sequence that you uploaded and the genome annotation file that you uploaded. And if you're working with a more commonly studied species, then a lot of those genomes are already going to be available in the discovery environment at the genome services or in the genome services folder. So many plants, many human or many mammalian systems, all those genomes are already present. Okay, so GFFread, all of this is doing is creating a FASTA sequence file of all the identified genes or all the known genes in your genome from the reference annotation, and that is going to provide you kind of a reference to map all of your nanopore mRNA sequences back to. Okay, and then once you have that reference transcript file, then you can start mapping your nanopore reads to the transcriptome using the MiniMap2 app. So let's switch screens, and we're going to go through some basics of the discovery environment, and we're going to talk about how you can easily find apps and do some of these analyses. Okay, so you should be able to see the discovery environment. And for those of you who have never been to the discovery environment or seen the interface, it's very intuitive. It looks just like your computer if you have a very clean and uncluttered desktop. So unlike me. But up in the top left corner, you have 3 icons, data. This will take you to the data store where you have basically all of your files are located. You can access all of your files. I already have a tab down here where I have gone to the folder that all of this kind of example data, you can access the example data to process, assemble, and analyze some nanopore short read sequencing data, all on your own at a later date. But you see in this folder, I have some FASTQ files. I have the genes, so the transcriptome from the model plant species Arabidopsis thaliana. And then I have some output files from a couple of mapping programs, MiniMap2, which I briefly mentioned, and GraphMap, and then some output files some further output files that we'll talk about in a second. Okay, so that's, I mean, as you saw, the structure of folders and the discovery environments is very similar to if you were looking at your browser on your computer trying to find stuff. So very, very intuitive. In terms of searching for apps, again, also very intuitive. If you're good, we can search for MiniMap2, which is the app that I was just talking about, and click on it. And again, the interface provides a lot of the parameters that you would use in the if you were going to perform this in the command line. And you see here, there's little red asterisk next to things that you have to add. So of course you have to add sequence files, and you're going to have to add a reference transcriptome, etc. So very, like, the apps kind of walk you through things. So they're very, very easy to follow, very straightforward. And again, if you have any questions, you're confused about something, you can go down here to this little icon down in the bottom right, intercom. That will allow you to talk to people at CyVerse, the discovery environment team and the science analysts, and ask questions about how to use some of these apps. Okay, so let's go back to presentation, kind of finish walking through what you need to do to go from, again, go from nanopore sequencing reads all the way to some differential expression or genome visualization. Okay, so I mentioned MiniMap2. This is kind of the preferred program that Oxford Nanopore recommends to map your nanopore sequences back to a transcriptome. And when I'm doing this, I recommend, or when you do this, I would recommend launching 2 MiniMap jobs. One using the reference genome for your species of interest. So you just put that right in here in the reference genome FASTA spot. And then another with the reference transcriptome that we generated using GFFread in the previous slide. And the reference genome job is going to provide us with, is going to allow us to visualize our information in the EPIC-Koji environment, our genome browser. And then mapping back to the reference transcriptome is going to allow us to perform differential expression. And down here at the bottom are links to both the MiniMap2 GitHub repo and also read the docs, and then read the docs or the wiki for MiniMap2 in CyVerse's wiki page. Okay, after you map your reads to your reference transcriptome or your reference genome, even if you're using MiniMap2 or if you're using the alternative application GraphMap, both of these produce SAM files, which are human readable files containing the nanopore sequencing reads, the sequence of those reads, and where the coordinates of where they map back to in whatever transcriptome or genome you mapped back to. And unfortunately, the next application that you're going to use can't read a human readable file. It needs, instead of a SAM, it needs a BAM file. And so a binary file instead. And which means that we need to convert the SAM output from MiniMap2 to a sorted BAM file using the SAMtools app. Again, you can locate the SAMtools app by searching for SAMtools 1.7, et cetera. And then it's very straightforward. There's only one input file that you need to insert, which is going to be the input SAM file provided by MiniMap2. Okay, very straightforward. All we're doing is generating a file that our read counter can read. Okay, which goes to the next step, which is counting reads with the Salmon tool. So Salmon is one of the, I think there's a variety of read counting tools, which is basically asking how many of your nanopore reads mapped back to each different gene in your reference transcriptome, or to each different locus in your genome of interest. And so it's very straightforward. It's just counting. Yeah. And so Salmon is good because you can provide it with the reference transcriptome and then the sorted BAM file. Very easy to use. And all we're asking Salmon to do is tell us how many reads in that sorted BAM file we generated map back to the transcriptome we generated and we've been using to do all the mapping. Again, here are the read the docs for Salmon. There's only a couple of input requirements for the app in the discovery environments. That's the reference transcriptome that you generated with GFFread and the sorted BAM files that we generated in the last step. So next, typically at this step, so the output from Salmon is going to be a tab delimited file containing all the IDs of the genes in your reference transcriptome, and then numbers associated with how many reads mapped back to each locus. And typically you would download that table and you would go into your RStudio application on your computer, or if you like using R on the command line, you would go into the command line and start performing differential expression analyses, either using DESeq2 or using EdgeR. But thankfully, CyVerse provides us with the option to use RStudio in the cloud using these Vice apps. So these are visual interactive computing environments. You can find the RStudio Vice app in the discovery environment just by searching for RStudio DESeq2. This already will have all the libraries necessary to run DESeq2, all the prerequisites, so to speak, installed. And so it would pop open a new window in your internet browser. It would look just like RStudio on your computer, except you would be doing this all in the cloud. But the important thing is that you would have access to all the data files that you generated in this cloud environment. So everything that's on the data store, all the quantification files you generated from Salmon, you would find here, and you would be able to use, and you wouldn't have to do any kind of data wrangling, downloading, uploading, etc. So very intuitive. The Vice apps and the DE, there have been a couple of tutorials about them. I would highly recommend checking them out. They're incredibly useful. Okay, and then the very last thing is visualizing your nanopore data in Epic Koji. So Koji is a comparative genomics platform that contains a lot of useful tools for visualizing your data or performing comparative genomic analyses. If you've never visited Koji or used Koji, I would highly recommend checking it out. And one of the reasons I recommend using Koji is because Koji is directly linked to the discovery environment and to the data store, more importantly. So all of your files that are in the data store in the discovery environment, you can directly access them and upload them and move them into Koji's environment so that you can visualize your RNA sequencing data, your nanopore data, or what have you in the Epic Koji environment. And so very briefly, we're almost done here. I'm going to do one last share so you can see what I'm talking about. So we're back to the discovery environment, and you will all notice that so this is my main folder, and in your main folder, all of you, once you get into the discovery environment, will have a Koji data folder. And you can drop anything into this Koji data folder, and if you then go to genomevolution.org and you go to load an experiment, you will see that everything that was in your Koji data folder, particularly this nanopore folder that I'm highlighting here, is going to be easily accessible, directly accessible from Koji. And so you can easily load experiments, SAM files, BAM files, transcriptome files, etc., into Koji and visualize them in the Epic Koji genome browser. And what it looks like is something like this, where these are all long-read nanopore sequencing reads mapped back to Arabidopsis. And you can see the Arabidopsis gene, genome annotations, all these genes. And if I were to zoom in, which I don't really have time, you'd be able to see intron, exon boundaries, etc. Okay, so let's go back to the presentation one last time and finish up by saying it's as easy as that. And I would be more than happy to take any questions, and we can talk about email or Slack or what have you later if you have any additional questions. And thank you for your attention.
Andrew, thank you. I'm looking in the chat window and I don't see questions. You gave a great step-by-step, and thank you very much for showing exactly what the discovery environment looks like. If people have questions, I'll give a minute or so to type them in there. Otherwise, maybe Andrew, could you explain what you'll be doing at BTI going forward?
Yeah, so a couple of projects. One will be focusing on, well, I guess the overarching theme is that me and a couple of really amazing collaborators and kind of co-cluster hires will be looking at how plants respond and cope with stress. I think initially we'll be working with tomato and Arabidopsis species and close relatives. And yeah, doing some high throughput phenotyping, some kind of genomics and transcriptomics, and some really interesting protein work. So we have it's a really, really cool cluster of people. And we'll be doing a lot of different analyses, but mainly the big question is how plants cope with stress, and looking at kind of stress resilience and certain wild relatives.
Excellent, cool. That is excellent, and I'm sure the BTI folks already are familiar. One of them was an early collaborator with iPlant back in our iPlant days. So you won't be necessarily inoculating them to CyVerse, but hopefully we'll find you online then.
Yeah.
So great. I don't see any questions. So thank you very much, Andrew, and best wishes for your move to BTI. Everyone else, please join us on September 13th for a webinar called Genome Assembly using NanoDJ notebooks. The presenter is Tamara Hernandez Beftink, a doctoral student at the Universidad de La Laguna in Spain. She's one of the co-creators of NanoDJ, which is a Dockerized Jupyter notebook for interactive Oxford Nanopore MinION sequence manipulation and genome assembly. So that sounds great as a follow-on to Andrew's introduction. Thank you, everyone, and we'll see you online in a couple weeks.